I would be very glad if you took this QR code or this link and filled our questionnaire. It takes maximally about 20-30 minutes, but I think most people would have it finished much faster. And today we are having more like interesting public outreach day of this workshop of ours, focusing on astrobiological topics of Drake equation, searching for intelligent life in the universe, and what and the biases or shifts our thinking about these topics. We will start with Pieter's lesson on kind of introduction and how to communicate these topics, and kind of also art outreach, but also with some very concrete astrobiological experiments. Then we will continue with Julie's talk, she is just back there, about how to communicate astrobiology and astrobiological topics to public, and how tricky topic it is. And then we will have lunch break, and after lunch we will delve into some more deeper scientific aspects of this problem, looking on the quantitative side of the Drake equation, some of those factors, what we know about star formation or planet formation and planet types, and what we do not know, for example, about formation of life and evolution of life. And we will finish at 5 o'clock in the evening with Pieter's lesson, which will be kind of all-encompassing talk about the Drake equation, history of the Drake equation, searching for life, and also interactive part where you will be able to fill in the factors yourself and find out whether a Czech public, or at least Czech public today, is very pessimistic or optimistic about life in the universe. But now, without further ado, I will ask Pieter Steyaert to start with his talk. And yeah, he is an astrophysicist and artist from Copenhagen, so from Denmark. And you can start.
Thank you, Jan. Good morning, everyone. I don't think I'll give a lesson. I also don't think I'll give an actual introduction maybe to the Drake equation, even though I will touch upon it. But I'll try to ease us in some of the ways of thinking, and maybe with a concrete example that I think can open up the conversation nicely. So as Jan said, I'm both a visual artist and a scientist. My background is in computer science, but I ended up doing a PhD in, as part of an exoplanet and protoplanetary disk research group. And in the same way, when I studied art, first I studied graphic design, and then I went to do visual arts. So maybe there's a pattern there of me trying to shift back and forth all the time. I'm here to talk about a project that precedes both my doctoral research and that I also hope will extend for quite some time after it. I don't think I need to reiterate this because it was just brought up. But I just want to say, for me, the topic is not new, but the recontextualization of what I'm trying to say today is new. So I hope you bear with me while we're trying to do this experiment. It is always pleasurable to know who you are talking to. Of about half of the people who are sitting here, I have some idea. But for those who are sitting at the back, maybe, I'd like to know by raise of hands who of you knows or has heard, maybe let's say that first, of the Drake equation before you got invited to this talk today. All right, we've got one person. That's nice. And do you also have a general idea what it means? No? So you don't want to give it an attempt to explain it in your own words?
No, it just we heard about it in some presentation and some interesting topics, but I don't know about the explanation.
Yeah, okay. That's absolutely fine. I think especially later today after my talk, some of my colleagues will go much more in depth on different aspects of the Drake equation and also what those might mean. Jan has already mentioned this, but if you're like me, then you have a hard time focusing on one thing at a time, and I will be very unoffended if you fill in the questionnaire while I'm giving the talk. Feel free to do that. Also, if you're doing something else on your phones, I can have the illusion that you're just filling in the Drake equation survey that we've set up. Okay, so to have some kind of understanding and some type of introduction, I'll just say that the Drake equation is a probabilistic framework to estimate the number of advanced civilizations in our galaxy. And for me, and I think this is much more in line with what Frank Drake aimed for as well, it is a relevant foundation for a discussion, to have an engaging conversation, much more than it is a true equation in that sense of the word. It breaks down the steps that might be needed for intelligent life to emerge and to become detectable, and it's used in this to structure both scientific and speculative discussions about life beyond Earth, and it's exactly on this realm, this merging point between the speculative and the scientific, that I want to talk with you today. Jan's talk, especially, I think, will focus specifically at the end of the equation, the L, the length of the time that a civilization can emit detectable signs, and it is from another lens, something that I also want to focus on right now, maybe in a slightly experimental way, if you'll allow me. I want to talk about culture. I want to talk about the fraction of intelligent civilizations that develop communication rooted in ourselves, from our point of view as humanity. So therefore, I think it's important to talk about the Industrial Revolution. If we talk about this FC, I think it's important that we recognize that the Industrial Revolution, modernity, and its exponents later on laid the foundation of both intra- and extra-Earth communication through means of radio, technology, satellites, telecommunications emerging from industrial and post-industrial societies. The Industrial Revolution gave rise to modernity, which brought space closer to us, not only in that technological sense, but also the offset of these technological senses. For example, if we look through the lens of culture, here are some space illustrations from Frank R. Paul on the left of the screens, who was maybe the first major scientific pulp illustrator known for its covers of amazing stories. In his visualizations, you have space stations and rocket ship and alien landscapes that are really intertwined with the poetics and the aesthetics of the time by means of mechanical, pipe-laden metallics, and it's all rooted in this mechanical optimism in a very modernity type of way. It's a reflection of a society that understands steam power, steel, and combustion much more than anything else. So space gets expanded by being on Earth still, but the cultural expanse goes beyond our own atmosphere, which is certainly nothing new. However, this creative and cultural expansion of space imaginaries is not the only way of cultural expansion. For example, here you see on screen, albeit a bit small maybe, the Fallen Astronaut, which is a small aluminium sculpture placed on the moon by the Apollo 15 crew in 1971. And it is the first and possibly the only artwork on space until in a few weeks when we have a new artistic mission that is launching there. It was created by the Belgian artist Paul van Hooijdonk, and it lies on a plaque commemorating 14 astronauts and cosmonauts who gave their life in space-related missions. It was intended to be quiet, to be memorial, and not part of the official mission payload. However, and for this reason exactly, the NASA and the mission, the Apollo mission, agreed that it should remain anonymous and without the artist name attached. However, my countryman, Van Hooijdonk, he announced plans to sell replicas of the sculpture after it had been in space, which created controversy. It went both against NASA's policies of non-commercialization, and the Apollo 15 crew themselves also distanced themselves from the sale. These agreements followed, conflicts arose, and despite this controversy, it's still the only human-made sculpture that we know on the moon, unless you recognize the engineering or the flags as sculptures as well. So interesting here, for the get-go, the moment we ventured out in space is also the moment we had this conflict of human nature, of capitalism, arising at the same time of the first installment, what I think is significant to take into account. If we move on from this purely cultural lens and go back to a more industrial way of thinking, we have to recognize that space industry has been recognizing significantly. This graph visualizes the number of countries that is launching payloads in space, and it's increasing every year. I'm not sure if you can read it, but it starts in 1960 and it ends up 2 years ago, I think. It allows for a democratization, in a certain sense, of near space exploration, but it also expands and infuses our chemical atmosphere with a new silicon layer of space debris consistent of defunct space satellites, rocket stages, which are residing often in low Earth orbit. This democratization process also results in a hefty drop of price per kilogram and a payload for these LEO-bound spacecrafts. So it sparks, or it allows the spark of a commercial and a scientific space industry exploration. It expands the reach of humanity not only culturally, but also economically. So we're expanding once more humanity going beyond Earth on a technological level. And this line between what is on one hand maybe cultural, on the other hand fantastical, and then the hard economic or scientific divide, it's not always so clear. The image here in the center depicts a commercial graphic from Spaceborne United, a Dutch startup company that is actively seeking to conduct in vitro fertilization research in near zero gravity, because according to them, that is the crucial next step to humanity. And then on the right here, you see a portrayal of the O'Neill cylinder by the physicist Gerard K. O'Neill in 1976. It portrayed a modern utopia long after modernist has ended. Humans, we've had throughout different civilizations this deep fascination with creating things that will withstand the test of time. From both the pyramids in Egypt to buildings of the Roman Empire, the art of and architecture of Mayan empires or Hindu kingdoms, we've always been captivated by the idea of preserving, maintaining a legacy for the eternity of the culture or the individual that is de facto leading the culture as some kind of empire. And it's maybe the same desire for permanence that is driving our collective imagination as humanity to explore the vast and boundless areas of space. Many people view or have viewed the prospect of living in space as a pinnacle of human achievement and a testament to our ingenuity and technical prowess. However, this desire for an external existence from Earth can also be a source of conflict and tension, because it can lead to the belief of us versus them, that one culture might be superior to other and a drive to dominate other cultures. But this idea of the utopia, the modernist utopia, settlement in space away from the limitations of Earth, it's not something that we have forgotten, it's not something we have left behind in our imaginaries, and it's not only something that comes up in science fiction, literature, or visual art. For example, here you see quite a recent artist impression that was commissioned by the commercial space company Blue Origin, which resembles very much the graphic from O'Neill that we saw before. Obviously, this modernity and these utopian views are in quite sharp contrast of our lived experience and the reality that we're facing with our generation in the state of the world on Earth. One of the other outcomes of Industrial Revolution is a global climate change and dynamics that are rapidly changing in front of our own eyes. And this, for some humans, leads to both a philosophical and even a physical expression of escapism. So we're staying with the gorillas of Silicon Valley, but now shifting towards Elon Musk, who has quite often in a neoliberal USA as a hyper-capitalist been on the forefront of the second space race that is happening in front of our eyes. And in that context, he talks about Plan B, or the backup for civilization in case Earth becomes inhabitable. And if you're in doubt ever how ill-conceived this idea is, feel free to ask my colleague Nana, who's sitting here at the front, who can talk much about the habitability both within and outside our own solar system. So to summarize this first part of my presentation, the Industrial Revolution, modernity, postmodernity, hypermodernity, and so forth are all engines that are shaping cultural and capacity for communication on one hand, but at the same hand, it's also threatening the longevity that we can maybe conduct these communication. It introduced risks like climate change, nuclear threats, war, ecological collapse, and so on that could shorten this L. Therefore, it endangers, but also improves our visibility in the cosmos. All right, we're here, we've made it through this part, that's great. And I think it's maybe a good idea to leave all these big and slightly abstract ideas behind. It is Thursday morning, after all, and the weather is gloomy. So maybe we should go to the small and the tangible, and maybe the somewhat silly looking. So it's almost tangible. This is the rotifer, quite specifically the bdelloid rotifer. It's microscopic, it's aquatic, it's an animal, and it's commonly found in freshwater environments. And they may appear a little unremarkable, albeit funny. They possess, I think, quite fascinating characteristics. They're known for their unique biological structure, including their ability to survive in extreme conditions. We have known for individual organisms that have been about to be revived 25,000 years after being frozen in the permafrost. Furthermore, they reproduce through parthenogenesis, through asexual reproduction. And this is what Professor Van Doning describes as an evolutionary scandal. Because on one hand, how are you able to survive extreme conditions, while at the other hand, your main weapon that we have within life, sexual selection, is not there. We just clone all the time. So how do you bring those two aspects together of being really good at adapting yourself to changing conditions and on the other hand, not having a sexual selection framework? Furthermore, and this is maybe even more extreme, when they are being confronted with drying out, when the aquatic environment is drying up, they die in every sense of the word, I would say. The cell activity stops, and they go in a state of anhydrobiosis, where they can be in a dried-out stasis for many years. It lays there, it falls apart up until the cellular level, and their DNA starts to break apart as well. Until you rehydrate them, until you introduce a droplet of water. And I think this might have been one of the sources of inspiration for the three-body problem, if anybody has read the trilogy. But nobody has ever confirmed that. It just seems wild that there wouldn't be a connection. Anyway, when you introduce a drop of water to the rotifers, they start to restore their DNA and spring back to life. And it's exactly maybe in this phoenix-like behavior that the answer to what I called before the evolutionary scandal lies. It's during this DNA reparation phase where one of the mechanisms kicks in that we call non-homogeneous end joining, which basically just means that you're joining DNA fragments left and right without a fixed template. It's only one of the strategies, but it is the most crucial one for the story that we're telling. Because even though they're quite good at trying to avoid errors when they do this reassembling, they do absorb foreign DNA from bacteria, from fungi, from plants during this rehydration process. This foreign DNA can be incorporated in their genome and therefore be used to patch up regions that have been dying. And it's theorized that 10 up until 15% of the genome may come from other species. And this is what we call horizontal gene transfer. And it's maybe there that lies the answer to the question from before. How can they be so well adapted to changing conditions? And it is because they use this horizontal gene transfer, incorporating genetic material from their environment. And this has been a topic of research for Karin Van Doning. She's a professor that I mentioned before who coined the term evolutionary scandal, who's been working for decades on this particular, on the rotifer, on the bdelloid rotifer, and runs a lab at the ULB in Brussels, as well works together with her colleagues in UNAMUR on this specific animal. And she had a fascinating idea. She said, okay, these rotifers are good at restoring their DNA, which is wild. We've been working on this for many years by now. But what would happen if we bring them into space? Are they able to restore their DNA in the same way due to cosmic radiation as what happens when they're dried out on Earth? The impact of this research is, again, on the edge of the scientific and the speculative, because it could wildly change how we think about deep space travel. Instead of protecting ourselves from cosmic radiation, we could maybe just come up with ways to restore DNA and repair whatever is broken. On the other hand, it might give us insights on cancer research on Earth, for example. However, she wanted to expand this conversation and these experiments beyond the purely scientific and making sure that the general public was involved in this conversation as well, and therefore she invited a bunch of artists to join in the dissemination. And that's where we come in. So about 15 years ago, I co-founded a transdisciplinary collective called SEEDS, Space Ecologies, Art and Design, which has grown since then to be a global actor where we prototype around future thinking, often through the lens of art science, and we contribute both artistically and scientifically. We wanted to be part of this project, however, we just didn't only want to do dissemination, because most of us have a scientific background as well. We wanted to be part of the experiments as well that they were planning to conduct in space to see if these rotifers could restore their DNA. The slide that you see here is not our entire collective, it's just a subset of people that have been working since 2019, 2018 on this project. The artwork has two distinct aspects. One is purely conceptual, and the other one is rooted in science exploration. But the outcomes are also twofold. One is different media artworks, and the other one are scientific and academic contributions by means of academic article writing. Remember that O'Neill cylinder that we were looking at before. We thought that this was a relevant idea. We witnessed both contemporary and historical human civilizations, and we thought that their drive for eternity, cultural immortality, would be a good starting point to put next to the rotifer, exactly against the backdrop of their space explorations. What we wanted to do is use the contrast between stasis and flux as a metaphorical device. We want to critically reflect on the aspirations of humankind in space, raise questions about the concept of cultural and identity in relation to space, and use it as a vessel to consider who has something to say about this development of cultural and identity in space. Because although the democratization process is ongoing, it's still extremely privileged and shielded entities that can actively engage in space research. We want to explore both how cohesion can be maintained, while at the same time we can celebrate humanity's rich diversity across cultures and identities. And so in two articles, we have explored this conceptual theme. Engines of Eternity, which is the name of the project, uses these biological characteristics of the rotifer as a frame to question humanity's obsession with creating a perfect society, possibly in outer space. And it comes from this idea of cloning, right? Keeping the same idea forever and ever, keeping something for eternity, just like what cloning would do, which is in sharp contrast with the adaptability that we know in the horizontal gene transfer from the rotifers, just like our human desire for cultural immortality is in sharp contrast with a constantly evolving cultural fields. And so we coined the project in order to examine all of this through the smallest creatures in Earth, the rotifers. As I said, the concepts were not only explored throughout papers, but also through different artworks, for example, a video essay that talks purely about these conceptual big ideas that I've been throwing in your direction. On the other hand, it is just the backdrop of what we set out to do later on. So you've heard me say all these fancy words rooted both in science and in humanities, all these big concepts, and you can say all of that is fine and well, but what did you actually do? We were struggling with the same idea, especially since we wanted to be part of the mission going to the International Space Station. We thought it was important that the artwork would also physically be part of it. which we were allowed to do. The only problem is the only place that we got was on the mission bags that conducted the experiment. We got 1 square centimeter. And I just said a lot of fancy words that are quite hard to put on 1 square centimeter. And then the second constraint was that we could only use a Sharpie, which is not the best tool if you have to fill 1 square centimeter with a big concept. Yeah. Obviously, and I think it's important to recognize this for what is probably more a scientific crowd, there's a symbolic gesture that is attached to it that is much more than purely the physical action that is being taken. So things need to happen quite fast, and what we decided upon doing, also in secret, because technically we were not allowed to do this from the NASA in Cape Canaveral, we applied through a stencil representing the part of the DNA, but also the infinity symbol on 10 bags. So one of us put Sharpie on the fingers and through a stencil applied a fingerprint 10 times on the mission control bags, because it represents uniqueness, identity. The fingerprint is unique. As I said, it is more a symbolic gesture than a very deep artistic concept, because the artistic concept lays in sending it up to space and later on modifying it based on what happened in space. So we had 10 bags that were part of the payload going up to the International Space Station, each containing 10,000 rotifers, and then 10 bags that remained on Earth. So we had a control group on Earth, including the exact same rotifer societies. Then on board of the International Space Station, we had trained astronauts, in our case, Luca Parmitano and Michael Hopkins, conduct the experiments. And the first mission was quite simple. It was exploring the impact of near-zero gravity on protein expressions on the rotifer animals. You'll never know what we did. So, because we were actively asking to be part, maybe I'll just repeat what I just said because it might have got lost, but the first mission was focusing on the protein expressions in near-zero gravity across the 10 bags containing 10,000 rotifers each. As you can imagine, the purely scientific team in Unamur and at ULB, they were not super exciting that a bunch of artists were also going to be working with their data and claiming that they were conducting scientific experiments as well. So we tried to meet this initial skepticism by having a lot of conversations with the scientists. However, it's important to note here that we didn't ask them regarding the content of the protein expressions. In fact, for us it was crucial, and it's not unlike maybe SETI research, that we didn't know anything about the data that we got. We knew there were protein expressions. Within the team, we have people like me, computer scientists, or people who have done bioinformatic experiments before, but we didn't know anything about the rotifer. Obviously, we can look up, but you cannot cram a full PhD or full education over the course of a few weeks when we're doing this in our spare time. It's also maybe important to notice that this was done for a big part during the pandemic. So while I was locked up in a tiny apartment in Manila, the rocket was flying up in space with our space artwork and research happening, which was also quite a contrast. But we didn't know what these rotifers were, and what the protein expressions meant. So step one for us was to start exploring that, to build tools, to build visualization tools, drawing both upon our artistic backgrounds, our design backgrounds, and our scientific understanding to increase the knowledge of what it actually was that we were looking for. And we didn't look at what the scientific team, the purely scientific team, was doing because we didn't want to mimic what they were doing. For us, it was really important to create meaningful insights and not just using the data as some kind of noise or seed to generate an artwork that would only symbolically represent space. At the end of this whole exploration process on the protein expressions of the rotifers that have been to space, we landed on this final step. We developed data exploration tools focusing on geoterm analysis, which means gene ontology. We were establishing the hierarchy among different genes and the quantity of their expressed proteins, and then match these to gene ontologies. We were trying to map and visualize both functional and meaningful differences because we set these up against the mean average for each of these ontologies on the bags that are a control group that remained on Earth. And remember our fingerprints that we sent symbolically to the International Space Station? Here we stitched them together, and this is maybe the foundation of the artwork that I wanted to show in this schematic. We don't have enough pixels to actually read what is there, but I'll just guide you through it. We have the original fingerprint glyph that you see on the top, and then using the data that came from the first experiment, which was purely looking at this protein expression. You saw the visualizations before, right? These 8. You might ask, why are there 8 and not 10? It's because 2 bags we didn't analyze. So that's why they're white. And we used these visualizations in order to push and pull the original glyphs that were sent in space. They already have some kind of seed function because they're not identical, because the fingerprint would always make small differences, obviously. And that was exactly the essence. So when there is a lot of difference, it would pull the pixels together. When the difference was smaller, it would push them apart. And each region is clustered functionally. So for example, enzymes linked to morphology or enzymes linked to movement would be clustered together in a logical way so that the end result is also in a hierarchical way linked together that makes sense purely than using it as random. Then we stitch them together in a circle, and then I build an algorithm that extracts them into bigger sculpture, focusing on the largest differences towards the smaller differences, and these sculptures were then presented as sculptural artworks. They were 3D printed, but there was also, trust me, a lot of manual labor involved because they're quite large scale, and a lot of acid baths that were happening. Then for the next mission, we didn't use protein expressions anymore, but genetic mutations we looked at. And again, we applied the evolved glyphs to the new mission modules when we sent them up in space. Therefore, we're not the first artwork in space, but I think we're the first evolving artwork that is influenced actively by the environment of space. We're not only using space as an exhibition space to broaden our cultural expanse, but also as an actor that actively partakes in the work. So this non-human agency that we're trying to discuss is not only the Rotifer, but it also becomes space as an environment itself. For the last mission, the third mission, which was launched in 2024, so last year, this is data that we're still looking at. So we haven't built that sculpture yet, which is why you saw in the schematic it as an outline. We're looking at the actual movement of the rotifers in near zero gravity. And then we plan for a final mission, hopefully this year, where we bring them outside and then look actually at the cosmic radiations. As I said, it's not only scientific research that we do, but as you notice, there's a lot of artistic components, and then we do these exhibitions with them, obviously. Here you can see my colleague Ulrike looking at sculpture. And we incorporate various media, video essays, sculpture, drawings, space mission artifacts, wall prints, interviews with the scientists involved. So there is obviously also a dissemination component to it. It is science communication, except the essence of it might not be science communication. It evolves, it's reconfigured, because as you saw, we have different missions, so the artwork becomes different every time we have a mission, and then we exhibit them in different ways. We've had exhibitions so far in Copenhagen, in Germany, in Belgium, and in the United Kingdom, and maybe one day here in Czechia, who knows. I want to end with the following. The use of these different techniques to create the artwork, to understand the protein expressions, has led to different results compared to the purely scientific team at UNAMUR and at ULB. Obviously, one answer could be the artist made a mistake because they're only artists. However, we double-checked a few times, and we don't think that's it. What we think is that because we worked a priori, without any bias, without the same level of understanding, we had an opportunity to look at all different facets in a childlike manner almost, which might have uncovered different information that was disregarded from the purely scientific team, who had a much more understanding way of working. And it's exactly this friction between the two that we're exploring, whether the inclusion of these artistic practices in addition to the purely scientific practices could have led to new insights. Finally, on top of that, we're currently working on the data of the last mission. So this is software that I'm writing right now that is analyzing the patterns of the rotifers when they move in near zero gravity. And what I hope to do, if funding permits, is to look at not only, the scientific team is looking at velocity, acceleration, direction of the rotifer, but I think it would be interesting for us to go a little bit beyond and try to include, once again, these artistic practices. In this case, it might not be so much on the visualization, but what if we look at dance notation? Dance notation is something that is heavily used in different ways. In performative arts, it is like sheet music, but for bodies, for dancers. And I think it would be extremely interesting to have a look at the movement of the rotifers, not only in heuristics that are purely discrete, but also try to see if we can label and identify these movements in ways that go beyond, like in dance notation, with the help of machine learning algorithms. But that is something hopefully for the near future. So I thank you all for your attention. I hope that it in some way expanded your notions of both human and non-human culture in space. And I'd be very happy to take any questions you might have. However, this is also a good point to take into account that I'm not a biologist, and I think quite a few in the audience were or are, because we've lost half of them. Thank you.
Okay, amazing. Thank you very much for your talk. And now we can start with discussion. I will give you this small microphone so that we have a nice recording. Nikos?
No, I was thinking that there is no need for mic.
It's for the recording.
It's for the recording.
Ah, okay. Okay, thanks, Peter, for this nice talk, reminding us the cultural dimension, which sometimes we're tending to forget it. And it is very important, in fact. I have perhaps a naive question. In the end, about the rotifer. Of course, you said you are not a biologist. In the end, you mentioned the aspect of gravity, which is absent in space, so this is important. But the other thing you mentioned about the ADN and the effect, the possible effect of cosmic radiation. Now, there are two things there. The first one is that the space station is within the magnetic field of the Earth, the Van Allen zones, which protect the astronauts from the cosmic radiation. That's why they can stay up there for months. I mean, we would never send astronauts to the moon for months. It's impossible. So the radiation dose that we expect for the rotifer to have is small in principle. But there is another thing. I mean, such small doses of radiation can be also obtained on Earth if you have you don't need to have the CERN accelerator, which is too high energies. No, just a normal accelerator. Nuclear physicists are using every day. Even accelerators that are used for medical purposes to bombard cancer cells, for instance. So it could be easy to use such an accelerator to bombard the rotifers and see how the ADN responds to that. And it would be simpler, in fact, than going standing up there. I'm just asking for this particular ADN. For the gravity, it's another story.
I think it's a very fair question. Thankfully, I do have an answer for that. We have conducted these experiments on accelerators on Earth, and we do know that different levels of radiation are no problem to the rotifer. They can restore their DNA. In fact, the question, and maybe I should have presented it in a different way because I understand the confusion, would be the other way around. The question becomes, in near zero gravity, are they still able to have their DNA restored? Because that we don't know. And in that sense, it's important to conduct the experiment in near zero gravity on the International Space Station, because we have very reasonable reasons to suspect that they can handle the cosmic radiation quite well, because they can do the simulations of that on Earth quite well.
Okay.
Does that answer your question? Yeah.
Thanks.
Any other questions? Yeah.
I'm surprised you still have questions, Nana. You've seen this project many times before.
Yes, I have, and it's equally interesting every time, because you make me think in different ways compared to what I usually would, which I know is also one of the purposes of your research, is to open up the way we view space research, at least for the space researchers. Of course, you also have other motives, but that's one of the very interesting aspects of your work. So a question I thought of, I really like your comparison between horizontal gene transfer and cultural evolution, like this weird balance between wanting to stay stable or not change, like keep whatever makes us us, while still being adaptable, and that those two are often, you know, they are put up as contrast, but both of them are necessary to remain or to maintain ourselves. So my thought is, when you think about horizontal gene transfer and cultural evolution, do you also see the do you see the parallels between adapting to new cultures? Like, so when we adapt aspects from other cultures, do you see that as a parallel to horizontal gene transfer?
Yes, I think that's indeed the point that I was trying to make. So I'm quite happy that you asked the question, because it's not for everyone the most intuitive analog that we're trying to draw. But it's exactly that. It is humanity being in friction, not only historically, but if we just look at contemporary society, on cultural identity versus diversity, and celebrating diversity. Celebrating diversity on a global scale, but also maybe the struggles on a national or even a regional level. And many countries are struggling with that. Political powers are rising due to those frustrations of not being able to solve that issue. And I think what we are trying to do here is to push a little bit back and saying this idea of a cultural identity of a nation, not only is it archaic, it's just never been a real thing. Culture has always been prone to evolvement, have always been prone to assimilate other cultures that are visiting and that they're interacting with. That doesn't mean that every culture is homogeneous or that every culture is exactly the same, but they are made up of different influences along the way. And I think when this project was conceived, this is not something I often talk about, but it was exactly the moment that in my home country, we were trying to, the right-wing government was trying to establish a cultural identity canon. What are the artworks that defined what it means to be not Belgian, but Flemish, a region in Belgium, which is quite ridiculous since we've been a country for such a short time and the cultural history predates it much larger. So I think in that sense, as a Belgian, we're very prone to asking these questions because we lack this idea of a cultural identity. Thankfully, I've shown you our collaborators. There's not that many Belgians involved. We're quite a large international group of people. So that doesn't make it a Belgian artwork. But as a Belgian, I think it's part of the answer.
Any other questions?
I have another question.
Okay, maybe just
Yes.
and then
Were there someone else who was raising their hand?
maybe during that I have a question. Okay, Zira, you can.
I have just a very maybe simple, maybe the most complicated question.
Okay.
What is your personal opinion about the length of the L?
Yeah, it's a good question. I'm not sure if my answer is that interesting, but I'm skeptical. I'm not skeptical for life as a whole, but I think that the L of humanity might be quite short. So since N is 1 in our case, I'm skeptical in that way. But I'm not super hopeful about what's happening right now in the state of the world. But we should.
Okay, if I may. Yeah, maybe a very short commentary on. I had the same idea how interesting is comparing the horizontal gene transfer to some like cultural transfer, cultural cohabitation and such, and the parallels may go even deeper because the incorporation of these like foreign genes into their own genome, both in rotifers and tardigrades and other such bacteria do it quite often. It is not random, but it has its rules. At first, these genes are usually put in some non-very frequently used regions. I simplify it very much, but yeah. Among genes that are not so essential, like for testing, maybe finding whether they are compatible or not. But from the macroevolutionary scale, we know that lots of super important genes, in fact, came from this horizontal gene transfer. So there must be some like inner long-term dynamic how to reshuffle the genome. So this again might resemble some like cultural exchanges and identities and such.
Yeah, no, it's a very good point. I think we can push the metaphor even deeper. At one point it will break, obviously, because the metaphor serves a function, and in this case it is to explore and be creative with both the bioinformatical ideas and then the human culture ideas. But to build on top of your remark, you also have the non-random horizontal gene transfer happening, where they use part of redundant DNA as templates to restore the DNA that was broken before in other regions. So I think this redundancy of maybe the majority of your cultural identity versus the minority of the diversity groups, I mean, there's something there that we can continue talking about. So maybe you should join the project and introduce some of your ideas.
Maybe, and I'm sure my biosemiotic colleagues would really like this way of thinking about parallelism between like biological things and the social ones. And maybe a short question. I am not sure I completely understood those visualizations of proteomes. Maybe if you bring them back, these are kind of like distorted, right? And the clusters are based on the similar function of the genes, but the distortion was also affected by the fingerprint patterns. Did I understand it well?
Yeah, so actually the distortion happens later. This is the template, the source that will create the distortion. So obviously this is, for people in the know, it's a super B-tree visualization where the smallest parts are the individual gene ontologies, and we always build up higher until you reach less abstract and more understandable, let's say, functions. So you can somewhat see it. This is one region, the left corner, and then the right corner is another region. And they're just visualized in such a way because we needed to bring the three regions in one square for us to apply the actual pushing and pulling algorithm of the source code. But there's no transformation happening now. In fact, this is the thing that leads to the transformation. Does that answer your question?
Yeah, I think it's clear. And
so this is one gene ontology, for example. These dots, because it's black, it means that this particular component will have vastly different protein expressions compared to the average of Earth. And they're all different because all the backs are slightly different, but you can also see some similarities, right? For example, you see that in the right top corner, the big group cluster at the back is quite dark, so that means that there was definitely a significant impact of space for that region, which is movement, by the way. So it makes sense that there's impact there because they're in near zero G.
Yeah, and the fingerprint patterns was like a separate thing. It did not affect these visualizations.
No, so it's the other way around. It is the fingerprints, they evolve based on the squares. So imagine the squares that I just showed. You put each square on top of one of those fingerprints, and then the color of the pixel will push or pull. So the impact of the sequence will make it further or closer away from each other. But it's a good question. I think I need to incorporate that much more in this schematic. It's not often that people actually continue to ask about the schematical and the functional aspects of the artwork, because it goes over the head of a lot of people, I would say.
Yeah, so maybe I was focusing more on the functional thing, but it is still nice. It kind of carries on the information, although in some very artistic way.
Exactly, and that's the idea, right? That the information goes back into space and then gets influenced by that next mission and goes back into space. So that's why I claimed before that space becomes an active agent in the evolution of the artwork. What people see in the when they come to an exhibition, they see right? They see this big screens, big prints, sculptures, and it's quite impressive. But the actual artwork is just the fingerprints in space, the glyphs, the teeny tiny 1 square centimeter things. That is a lot more powerful, conceptually speaking, I would say, than any of the other outcomes of that we're presenting. Yeah.
Thank you.
Welcome.
Yeah.
I was really intrigued by your proposition to make a future dance performance based on Rotifer. And it got me thinking about how it could also include the passage of time, because it could show how still it can be for extremely long time periods in the state of anhydrobiosis. There could be the dancer, you know, crouching extremely still, not moving at all, and behind them like a montage of time passing by, of the earth changing, seasons changing. And after that, there can be depictions of water and, you know, the movement starts. I think that could be a really powerful image to accompany the exhibition, or, you know, just on its own.
This sounds really interesting. There was no plan to make a dance performance. So I might have miscommunicated. We want to use, or I want to use, dance notation to analyze the Rotifer. But that doesn't mean that the dance performance is not in the sky. In fact, SEEDS is an open collective. So people are always welcome to join us. And if you want to use us as a platform to build upon your ideas, for example, to develop a dance performance, you're very welcome to reach out. Obviously, Julie, you already have quite some friends that are working with us. Yeah, that sounds really interesting.
Thank you very much. And I'm afraid we have ended this so that we can switch to presentations. But thank you again.